Education process. Okay, in our studies, and we are always open to hear back from the community and integrate your ideas into our yearly plans. So please feel also free to communicate with us if you have any ideas related to these issues. The committee is now co-chaired by Trevor, Nolan, and I. It was previously chaired by Ken Birnbaum, so I want to thank you, Ken, for doing this before us, and thank all the members of the committee, which you can see here on the screen, for putting together the wonderful list of speakers for this series. I want to now hand it over to Trevor to give us a little bit more information about this webinar series that we're going to have this year.
All right, thanks, Natanella. I'm Trevor Nolan. I'm an assistant professor at Caltech, and I'm going to start just by giving a bit of background about why we're having this series and the kind of questions that we're thinking about. So in the Plant Cell Atlas, we've heard several amazing webinars about single cell, about spatial biology, all these new technologies that are really changing how we can answer this question of what is a cell type? And here I have a couple different perspectives of discussion of this in human and animal fields, and there's been a lot of really interesting perspectives on this. You know, we've gone from thinking about just morphology and function to actually the molecules inside of cells, but the question is really much more complex than it might seem at first glance. So you might say something like Italian Eye says here, if you just tell me what genes are turned on, I can tell you what cell type it is. But perhaps there's even more to it than that. So to understand cells, maybe we need to start thinking of them as a cell tree rather than just a cell reference. And moreover, you really need to understand how a cell responds to really know who it is, as exemplified in this other perspective. But of course, plants have unique characteristics and properties, and so this means that the definitions that one might come up with might need to be tuned or have more nuance in the system. So we want to understand, what is a plant cell type? Should it be related to anatomy, function, lineage, molecular content? And moreover, because plants exhibit remarkable plasticity, responding to their environment or even regenerating whole plants from single cells, how do we define the plant cell states, for example, in different phases of the cell cycle or different environments? And how do we incorporate this with new technologies that we can use to profile these? So there's already been a lot of exciting discussion and writing about this, and I think that there are two pieces that I'm going to mention here that really nicely summarize the state of this, and that is that, as our own committee member Zahan says, this is rapidly evolving how we're defining this. And as this perspective from Adrian Rowe's lab mentions, it's complicated. So I encourage you to take a look at these and to participate in the discussion. We're going to have a really exciting lineup of speakers that are going to take us all the way from cell identity across species to how cell identities are acquired, how we can analyze them computationally, how we can compare them and look across evolution, and also how we can go beyond just thinking about RNAs and looking at proteins, metabolites, how we can look at the epigenome and see how it's changing our view of cell types and cell states. We'll also look at examples that may challenge our traditional views of these, like a single cell that has different functions, and think about how this is changing across different environments. So we want to open up this series by asking your thoughts on how are you currently thinking about plant cell types and cell states? How could we better define these in the future? So if you scan this QR code here, it will open up a Padlet where you can add your thoughts and ideas about this. And I'm also going to put the link to this in the chat right here. So We want to do this at the beginning so that we can get a sense of where we're starting at, and also so we can look as we go throughout this series and think about how our thoughts and ideas are changing, and how we can generate new and exciting directions and come together as a community to better define these terms, and to really push our research forward as the Plant Cell Atlas. So with that, I'm going to pass it back to Natanella, who's going to introduce our first speaker.
Yes, so it is my great pleasure to introduce Dr. Dennis Stevenson today. Dennis is the Vice President of Science and Coleman Senior Curator at the New York Botanical Garden. He also holds several adjacent positions at Yale University, Cornell, the University of Napoli in Italy, and others. His major research interests in the past few years have focused on the evolution and classification of psychedels and their placement in seed plant phylogeny, including the publication of the textbook, Plant Anatomy: An Applied Approach. As part of his field program, live plants are collected for laboratory research and for breeding purposes in order to save the cycads from extinction. So Dennis is part of the Plant Genomic Consortium as well, and is working to generate phylogenetic hypotheses on the origin and classification of the monocots. So I'm very excited to hear your talk today, Dennis. Please take it away.
We're working on it here. There we go, I think.
Yes, well, we can see.
Is that my calendar you see?
Yes, we see our calendar.
There we go.
Yes, here it is.
Okay. Okay, this is great. I'm really pleased that you invited me to do this, because putting this together made me do a lot of thinking about some things that I haven't thought about for a while, and I think that was good for me. In a context for me, as I started out as a plant anatomist, basically a developmental plant anatomist in my career. And what's happened for me is the techniques have changed dramatically. And so this first slide here, I'm just sort of showing, I've gone from using machetes to doing in situs. And when I started out, basically the new tool was transmission electron microscopy. And then, we got SEMs, et cetera. So, and in fact, TC scanning too. That's just one frame from a TC scan, where you can actually look at the three-dimensional structure of like a vascular system without having to do clearings and go through all that stuff. Anyway, let's take a look here. Would be nice to advance. There we go. Okay, so I'd like to put this in a little bit of a context. So we now more or less refer to plant anatomy and morphology together as structural botany. And I look at that as part of a continuum here, where we need to be more eclectic and sort of integrate ourselves with many, many other areas. And by doing so, I think we reach common goals we have, such as I mentioned here, conservation, agriculture, various other things, by just employing a lot of different information and analyzing it together. One of the things that made this talk possible are these sites. So I can send these to people, but the plant ontology, I was the PI on the 2 or 3 NSF grants for the plant ontology, where we tried to develop an anatomical morphological ontology that we could integrate with the gene ontology. Also, for any of you that need them, there's a place called CUPAC, the Plant Anatomy in Images. You'll see most of the images I used actually came from that site. And there's a related site that was part of that, but now is at Southern Illinois, on images of whole plants, there's thousands of them, and they're really excellent quality. So a little bit of background. I mean, plant anatomy's been around for a long time, and it's gone through some changes in those time frames. It started out mainly as a descriptive science, where people just wanted to describe the structure of the plants, and some of that probably even goes back to people like Theophrastus, who knows. And then in the latter part of the 19th century, or the middle to latter part, we really went into a functional phase where people were trying to relate these structures they saw to functions of some sort. And that, of course, then led to the developmental phase, which was really what was big when I came through as a graduate student, was cell development, but also tissue and organ development. And then when that sort of got put together with an evolution phase, so that we're now, in my opinion, in structural botany, really in the evo-devo phase. And to me, that's very exciting. Now, structure, though, we need the typology and we need the descriptors for the structures, and they have usefulness. I'll show you one in paleobotany. Also for systematists that are trying to do phylogenetic analyses so they can code characters. For the physiologists, geneticists, and archaeology, and that's what's on the right. This is called the bog of serpents, and I've done a lot of plant anatomy identifications for people in the humanities. In this case, we needed to know, because it had something to do with a court case, but we needed to know what was the species this was constructed from, what was the size of the wood that it was used, what was the age of that wood. Without having growth rings, we were able to do that. So that's just a different interface for plant anatomy. For paleobotany, if we look here, the top row are fossils and the bottom row are living cycads. And you can see the direct correspondence here between the inverted omega pattern of the leaf traces in the living one to that in the fossil, or this peculiar polycentric growth system, or the mucilage canals running through the rays. So my point with this is the paleobotanists need us to have a good lexicon, but also this gives us a time when some of these characters may have occurred in history. So we get a time frame and a structural frame. So as I started out, typology was the big movement, particularly in Germany. So there are type contexts. And for me, this is part of a context. In other words, we have cells and we have cell types, just a few there. Cells as a group together make tissues. And of course, you'll see some of the tissues have the same name. But the tissues then put together make organs, and organs become organisms. So we really want to think about organisms. This is just a typological diagram from Troll, but it's a useful construct for us in terms of the seed plants, or I should say the vascular plants. Well, that's a type context, but there's also a developmental context, in my opinion, in plant anatomy. The one based around meristematic issues, cell lineages, differentiation, what we call redifferentiation, and dedifferentiation, which is a word I hate, but I put it in here, and I'll explain that in a moment. And then there's those of us who really are more concerned with the functional context, both the function in a structural sense and a physiological sense, and both together in terms of the organism. We have primary and secondary issues, that is what we call primary walls and secondary walls within cells, but we also have primary growth and secondary growth dealing with meristematic issues. Totipotency is something I just want to briefly address. I love totipotency because any living plant cell is totipotent. It can become anything else. So what happens if you're a parenchyma cell and you differentiate, say, into a chlorenchyma cell, and then you lose your plastids, whatever happens, you become parenchymatous again. You don't retrace the steps. There's no dedifferentiation. It's a redifferentiation. It means differentiation is continuing, and we need to think about it as a continuum. The one that really gets me on totipotency, though, I have to tell you, is about the chlorenchyma. Why is it that roots are never green except in orchids? But take a robinia plant, a black locust. They make root sprouts on the farm. They used to come up. My dad would dig them out. A cell in there has its totipotency and gives rise to a whole plant that's green, but the root never turns green. So there's some interesting controls that go on in this anatomical world. Let me briefly talk about the enchymas. So we have parenchyma, which is a basic thing with totipotency. You put some chloroplasts in it and you've got chlorenchyma. Collenchyma is a what'd I do? a chlorhynchoma is a structural thing. We'll notice that the primary wall is thickened in the corners, the common corners. So this is just a primary wall, but it has some structural significance, presumably, in strengthening the cortex. Sclerenchyma itself is a tissue type, and basically, I keep doing this. I'm using the wrong thing. Okay, it basically turns into these thick-walled, secondary-walled cells in pairs. They're called stone cells. I have no idea what they do except make them gritty. But sclerenchyma comes in shapes like this, where it forms a tissue of sclerids that are more or less isodiametric and structurally significant. And then they often occur around vascular bundles. We'll see some of this later. And when they're like this as a bundle sheath, they're really elongated fibers. So in a certain sense, you have sclerenchyma cell types, some of which we don't know the function of them. For example, these things called astrosclerids, probably means you don't want to eat whatever that is. Macrosclerids actually form very hard tissues, for example, in seed coats, in structures like that. These are structurally significant, the osteos in like flimsy leaves. And finally, there are actually hairs that are called trichosclerids that become sclerified. In other words, they become lignified. The things that aren't usually included are fibers, but fibers are the same. They're very elongated sclerids, and they're structurally significant, and they actually occur in all organs and tissue types. We have the vascular tissue as a tissue, and in this case, I often include fibers in some parenchyma. So vascular tissue, we usually describe it as a primary system, which is basically your vascular bundles, develops from procambium, and then will contain at least two cell types, phloem, that'll either be sieve cells or sieve tubes, and xylem, which will be either tracheids or vessels. And I put in fern steeler types just because you should know there is a lexicon for the types of vasculature organization in ferns, and it's complicated, difficult, and scary, but it does exist out there. And then another important point of vascular bundles are often called open bundles. So if you look down there, these bundle types like amphicribal and amphivasal and all that, they all have xylem and phloem, but some of them have a cambium, the white line. That's called a fascicular cambium. So a bundle that's with fascicular cambium is termed an open bundle. If you have no fascicular cambium, like in the others, it's called a closed bundle. Let's take a look at the secondary part of this, because once you have the vascular cambium, you have your ring of bundles, and you develop an interfascicular cambium from parenchyma cells, and you get a vascular cambium. And that's going to give you secondary wood. Internal derivatives, of which are maybe 70% or more, will become trachary elements of some sort, either tracheids or vessel elements. And the outer derivatives will become the secondary phloem, or conducting cells for photosynthase. Within that, you may get sclerids and fibers and sclerenchyma. And if you look at the bottom panel here, you get living parenchyma cells in here amongst these trachary elements, well, which are dead. Well, these living cells are able to actually expand and contract with water conditions and actually help form a structural significance in terms of a standing tree. The phylogen over here produces what we call cork, and it actually is bidirectional and produces something called phyloderm, which is the outer part that you see as the bark on the tree, and an inner stuff called a phellum. So we have terminology for those cell types based on their derivation from a phylogen. I had this thought the other day, so I wanted to put this in here. When you talk about wood and all this vascular, look at this oak tree versus this pine tree. Look at the numbers of cells in the tracheids here that could occupy this. What I'm making the point is these guys are much better carbon sequesters than these guys. It means the guys with xylem that looks more like this actually would be a better street tree in terms of a carbon sequester. So it's just an interesting test. I should also point out that there are often resin canals, like in the conifers here, but you also get secretory canals in the wood of things, not oaks, but other of these flowering plants. So there are these different kinds of parenchyma cells that do differentiate, in this case, to be making a product that's secreted. Well, if we take a look at the ideal plant we had, and we get a lot of morphological changes that have occurred through time and in taxonomic groups, all the way over to things like these cacti. But what do they all have in common? They all have in common the same organs, and they're going to have all the same cells and tissues. But in this case, we sort of lost our leaves, but we turned our cortex into nothing but chlorenchyma and parenchyma. So we've just accentuated one and decreased another. But all the cell types are going to be in these kind of plants just like they are in these kinds of plants. So they're going to be ubiquitous. Another thing that I thought we should think about in this, and the reason I put this in, is that I want to put genealogy into this. And so we really need to think about how different lineages or genealogies have found different solutions to the same problem. So how can we capitalize on that? And perhaps they may have even found the same solution, but different genes and by different pathways. So that's just something for us, I think, to think about. So that leads me to this: evolution is about problem solving. So often you get the same answers. Sometimes the answers are different, and sometimes they're different but the same. What do I mean by that? It may be you develop a water conducting tissue that is actually identical in the way it looks, but different in its development or its origin. So when you have these things that look the same, but you don't know how they came about, it's really called homoplasia in a phylogenetic tree. The same structure in two different places, not identifying something. In other words, it's ignorance about the history, and it doesn't really exist. And as I say here, an A is not always the same A. What do I mean by that? Well, take a look at gene sequences. These are chloroplast genomes, but all I want to say is these numbers occur in position 830 is a thymidine there and here, but in all the groups, it's something different in between. Therefore, even though the thymidine molecules are identical, they're not the same ones because it's a different history based on the genealogy of the organisms. So it would be same for all of this. So this is just sort of my way of demonstrating those things. So we may have trouble in assessing these morphological characters, coding them in a data matrix, but they lend themselves to being tested because once you have the phylogenetic tree, then you can test how it became that way. My example here is perisperm. Perisperm is a different type of storage tissue found in the seeds of some of the monocots. It basically replaces endosperm. Well, it ends up that perisperm shows up as a homoplasia in different lineages on the tree of the monocots. Well, but if you go back and look at the differentiation of the cells and the actual cells they are derived from, it's different in these two taxa, Acorus and Natanella. Therefore, they're not the same thing, right? But they're not equivalent, they're not homologous, but we're stuck with peristerm as a descriptor for a character, for something we visually see but can't distinguish a difference. That's what we have to work on here, I think, in our terminology. Another thing is reversals. So sometimes on a phylogenetic tree, something will be present, disappear, and appear again in the tree when you plot it on there. Well, it can't be the same thing. And why do I say that? Because it was something else for a while, and then it's changed back to what it was originally somehow. So it's not the same structure at one level, but because of its history. In other words, it doesn't occur in nature because the lineages are independent, homoplasia. So the whole organisms evolve, and therefore the parts have to be coherent together. You can't have a different history of the pollen in a plant and the vessel elements in the plant. That is within that lineage. They all have to go together and have changed at the same time. Now, some things may change and other things may not, but it's a coordinated whole to give you a whole organism. So this gives us the idea that we need more robust hypotheses. Well, we need that by gathering different kinds of data. So my idea here is in the anatomical world, we need more information from Evo-Devo and comparative genomics. And that will allow us to choose from amongst competing hypotheses we might have. So the phylogenetic trees will point us to the way to getting the answers, but they don't provide them. The other thing I'd say, as I mentioned at the beginning, it's really useful sometimes to have the fossils to give you an idea for the minimum ages of structures, which may give you some concept on the idea of how genes have changed through time, assuming that that's what was going on in the fossil. Well, let me give you a couple of examples of these kinds of things. And one of them is the independent origin of vascular tissues, it says here, in Plitricaceous mosses. Now, in mosses, the Plitricaceous mosses, there's a center steel-like area that contains water-conducting cells in the center that are termed hydroids, and assimilate-conducting cells surrounding that are called leptoids along here. In other words, this is a higher mag, but these are the hydroids and the leptoids. Here they are in an ultrastructural point of view. So here's a leptoid, and one of the things, what happens to my thingy, is that when leptoids differentiate, they exclude or extrude the nucleolus from the nucleus. The nucleus breaks down, all the organelles break down, and the cytoplasm turns to proteinaceous slime. And if you wound it, it forms these slime plugs that plug the pores like you'd have in sieve tube elements, but in these so-called leptoids. They even have a callus formation in the walls. Well, here's the sieve tube element from an angiosperm with slime plugs, cytoplasm that is disintegrating into proteinaceous slime. These guys are identically developmentally, but they're occurring in different lineages. Consequently, we have these names hydroids and leptoids for the bryophytes because people have said, well, they're a gametophyte. They can't have the same tissues as a sporophyte and the seed plants. Fallacious thinking, but the terminology is good because even though it adds something for us, it gives us some better information. The same thing's true with these things. We have these vessel elements here with these sieve plates, often with simple ones. Here's the hydroids with these sort of thick walls, and look, some of them have big holes in their end walls. These are known to conduct water. These are known to conduct photosynthase. Other example that I'd like to use here is the independent origin of what we call vessels. Vessels is the water conducting parts of the wood. And we have here basically they're derived from what we call tracheids. Tracheids have imperforate end walls, that is, no holes in them, whereas vessel elements have holes. It may be scleriform, like this with many, or actually a simple vessel, which is probably the best. And this is water conducting, like we see the simple ones here. Well, there are at least 8 independent origins of vessels in the land plants. And they occur over here in Selaginellas, equisetums, some ferns independently, and then independently in gymnos in the Natanella, and then in the flowering plants. And in just some flowering plants, the primitive ones don't have them. So, but yet you can't tell them apart. I could put pictures of a simple perforation plates that are in the ferns, and it would look identical here. So they look identical, but we know they have different histories. This means maybe it's a good idea for us to go back and begin to look at how these things differentiate. Are they co-opting the exact same genes to do this from the general genome, or are they using different ones? You know, it's just interesting problems to look at in the development and differentiation of these cell types. I will point out one other thing. Tracheids, because they have no end walls, you know, if you wound a conifer, because they have no vessels, then they get embolisms in these trachea elements, which means the air bubble goes to the top here and no water can go through this continuous column of them. One of the things that vessel elements allow for, particularly these, is for embolisms to be induced and to go all the way to the end of the procambial cells. So you still have your water conduction functioning, at least in a better sense. Another one here that I wanted to bring up about this with these parallelisms is what we call a renchema. Now, it's a general term for these big cavities that can occur in stems, leaves, roots, wherever you have them. And they actually come in different forms. We don't know too much about what underlies this, I think, in an evo-devo sense, but we do know from developmental anatomy. So there are what are called lysogenous ones, and that's like in this equisetum cortex. This big cavity forms by cells lysing, and you can identify them because you have little bits of the cell walls projecting into this lumen that's created. That's in contrast to what we call the schizogenous, which occurs here in the equisetum plant separately, and in other many aquatic monocots, where schizogeny means the cells just separate from each other, and they do it in a very patterned way. And of course, you don't see any walls protruding because there's no cell death involved in this. And then finally, there's a third type called stellate parenchyma. Here's a parenchyma cell. Here's a branch from it. Here's another one. It's coming out in a common cell wall. So they look like stars or whatever. I don't know how much is known about the development of these, but anyway, we should. And then finally, I'd like to point out this one. This is a section of a bald cypress or the swamp cypress, and it will develop a renchema from lysogeny. You can sort of see bits of cell walls going on here in its cortex and perhaps in the secondary phloem, in roots and stems, and even in the leaves sometimes. However, they only form these if they're growing in water, like in the cypress swamps. If they're growing in your front yard and it's not a flooded area, they will not get parenchyma. So this means that the plant can respond anatomically, an individual, to its growing conditions, which I find rather fascinating, and I think an area that's worthy of research. Now, I want to bring this up because this is part of the plant ontology and use it as an example. These are the reasons why you want to cautiously handle your plant. And in the ontology, there was a word called spine, and a word called thorn, and a word called prickles. Well, they're not all the same. As you can see here, we have definitions for each of these. So what we did is we said, it'd be more precise and more interesting to people doing plant development and writing descriptions to actually use what these structures were derived from. So here's the examples. This is a leaf, and the petiole abscisses longitudinally and is woody to give a petiole spine. Hence, we use the term petiole spine. In some plants, their leaves will have stipules, and they become spines, like in this legume. These are the leaflets, but these are the two stipules at the base that get big and woody. Here's an example in a compound leaf where leaflets become spines. Here in cacti, they don't ever have leaves anymore. They just have that green flat trunk or round trunk. And those little things in the areoles there are modified leaves, total leaves that turn into spines. On the other hand, there are things called prickles. For example, on a rose, this is just a proliferation of cork that's full of sclerenchyma. And the third way to do this kind of thing is like in black locust, or honey locust in particular, is to take a whole branch, a whole bud that grows out, and turn that into a sclerified object. And one of the reasons you know that, if you could look at these on the top, they actually have a phyllotaxis to them. So there's vestigial leaf primordia and then axillary buds, right? So this is a set of branches as a thorn. So there are multiple ways to do this. And we now know in the plant ontology that we can use the ontology to tell people what it is they have. So I'm suggesting that that's what we're going to need to do with things like the vessel elements. They all look identical to each other, but we know they're independently derived. So it would behoove us, I think, to probably have an adjective on some of these cell types so that that immediately gives us more information about how it developed. Not only where it is in the plant kingdom, but possibly what genes were involved in that. So I think I will end there and just tell you that my favorite quote in the world is the one by Jacob Gronowski. I think that's the way we really advance science. The other one is you guys need to take a look at uh, this website. It's really nice. So I want to thank you for your patience, and I look forward to the rest of this series. Thank you.
Thank you so much, Dennis. That was super interesting. I could listen to this for hours, and beautiful, beautiful images.
Go
I'll,
to Kupak.
what?
Go to Kupak.
Yeah,
Those
yeah.
images,
We
90% of them were from that site, both the organism ones at the end and all those sections. Yeah.
I will definitely look into that, and we'll also share it with everybody that participated today. While Trevor looks at the Q&A box, and this is time for your questions, so I will prioritize other people's questions, but I have a question of my own, but if you'll be willing to stay a few minutes overtime, if we don't have time for that, I would love to ask it. Before that, I just want to ask you one of the three general questions we're trying to talk about here today, which is actually, why is it important? I just feel like you'll have a good answer. Why is it important to define cell type? First and foremost, before we
No,
actually start working at it, why do we need to?
there's multiple reasons for that. One reason we need to have them defined so we can identify them in plants, but we also need it because we need to understand the function and we need to understand how they differentiate it. So I think those are three different categories where different people have a need for it. You know,
And they
yeah,
also, it's when we define them, we can better understand the functionality. When we understand the function, we can use that to define, right? So it goes kind of both ways here.
well, and it tells us also like places where that organism might be able to grow or not grow
Yeah,
because of what it has and doesn't have. I mean, yeah.
Trevor,
Right. I love how you highlighted the plasticity in some of these features. I think that's fascinating to think about what it is, but also when it arises.
I love the idea that things get reinvented or parallel invented, out of that genome, that,
Mm-hmm.
which says something about how important they are for the plant,
yes,
right?
good point.
I think that's how I see it. Trevor, you want to start with participant questions?
Yeah, so one of the first questions that rolled in was asking about in plant cells, how do we define a stem cell, or what functions as a stem cell?
Okay. I mean, I know how I would define it. A stem cell is the one, well, that's interesting. I was going to say the totipotent one, but of course that doesn't work because all cells maintain their totipotency in a sense. But I would say, actually, for me, it has to do with the meristems. To me, those are stem cells. So, and it's called meristem, but I meant we have, I didn't mention all, but for example, we have marginal meristems in leaves and what are defined as submarginal meristems. And there's adaxial meristems to get round leaves, like in monocot leaf tips. So I think, yeah, stem cell I like to think of them as actively dividing cells, kind of, I guess.
How about like undifferentiated? What would you say? Maybe?
Well, yeah, undifferentiated because actually a parenchyma cell is differentiated because it becomes vacuolate and all that. That's probably good. An undifferentiated Yeah,
poised to give rise to particular cell type. Yeah.
undifferentiated totipotent cell.
Yeah.
There's another question. This says, lovely talk. What are the possibilities for cells with identical anatomy and unique evolutionary history? For example, reversion of a gain trait in one organ. And what would you think about that? Similar anatomy in the same plant, but are they the same cell type?
I think that's what I was getting at in many ways. Sometimes we need the same cell type as a descriptor, but remember I said we need some adjectives, or maybe we need to change them. And my real example of that is the leptoids versus sieve elements. In fact, they're sieve tube elements, there's absolutely no difference in their ontogeny and what they do, but they're in these two different lineages and two different parts of life cycles. And so by having the term leptoid, you sort of have solved that problem. The vessel element one is one where I don't know what you do with it, because if you try to name it for every lineage it turns into, you make a very encumbered terminology.
Sure.
Yeah, how do you go
Speaking
about
the same
that?
language is important.
Yeah, but yet we need the descriptors, right? So
Yes, absolutely.
it's typology.
Another question that Mohammed Shahid asked, and I think you had an example for that, is how is cell structure related to cell function?
Well, yes. I mean, think about this a little bit. There's I can't think of a better way a plant could get to move water through itself without having trachary elements. To do that, they have to be dead, but yet they have to have a rigidity to them, right? That's one way to think about it. So the structure function is important. Then there's the whole thing about, I mean, it's amazing really when you think about it, but how vascular cambium probably originated at least 3 times in land plants, and they all function the same way. So there's a necessity on the function side, isn't there? I mean, if it's not functional, it's not going to be here. That might be another way to look at it. Your history.
I wonder if they all acted the same and they're defined as cambium because they all act the same, like the functionality,
Yes.
or they were defined differently and yet they act the same. You know what I mean?
Yeah, I do. Hydroids and trachary elements. Good example. We use a different word, but they do the same thing. So yeah, we can get ourselves into a circle here.
Sure.
But it's curious to me because why don't we have cell types that are defined in fossils, unsuccessful cell types?
That didn't, that weren't selected.
Yeah, right. They're gone.
Oh, that's interesting.
Where are they?
That's a very interesting question.
Unsuccessful experiments.
Yeah.
Well, but I think it's one where we could think about what could possibly be a cell type that's not described, and then make a description, even though it's never been seen, and then look for it.
Super interesting, actually. Yeah. And even if you don't, so even if it's really lost, or maybe that what you meant, look for them in fossils. But so even if it didn't continue, why did it fail? Now that we understand what the cell type is, why did it fail? And maybe if we basically identify this, not novel, but ancient cell types that were lost, maybe at the environment that they were lost in, we could find some things that would be relevant in extreme environments now. I don't know, like maybe we can create them theoretically, if we understand the anatomy function, anything about them, that would be a very interesting, I think, scientific
I
journey.
could tell you one unsuccessful growth pattern type in a certain sense, dichotomous branching. You
Mhm.
can't have dichotomously big branching things except in cycads that obviously aren't dominating the landscape in a couple of palms because you can't support things that are dichotomously branched. They're just going to fall apart
Mechanistically,
weight-wise and
or
everything. So
mechanically.
the axillary branching helps solve that problem and making them all diminutive.
So how does it work in cycads?
They sometimes fall apart.
Okay.
Kind of like, saguaros, where one of those big branches is falling.
Yeah.
Yeah. Yeah, it's not a good life choice.
Yeah.
There's another question from Maria. I'm really glad to hear presentations like this. Do you think that genes that are involved in cell identity could be related to those involved in response to environmental changes? That sort of gets at this that we're discussing.
Yes, I think that's what I was trying to allude to in that bald cypress example.
Mm-hmm.
You're forming Aeronca is a tissue type, but you're forming Aeronca based on your environmental situation. So it's a what do you call it? It's a programmed cell death
Mm-hmm.
under
In fact,
certain.
the lack of forming lack of a
Yeah,
cell. Or,
well, to form those lysogenous, they have to kill a bunch of cells,
mm-hmm.
but they only do it under a certain circumstance, which means the program's in there, but it's only happens.
Right, in the right situation. Mm-hmm.
Yeah,
So how do you
kind of like
control
not
it?
turning
How to
green in the
regulate it?
dark.
Yeah,
So I
exactly.
think
That's
she's correct. Yeah.
There's another question here from Chris Georg Kunemund. I hope I didn't mispronounce your name. Is it common that the development of the cell identities goes wrong? For example, thorns of a rose growing inward or phylogeny in the wrong place?
I don't know of any examples like that. That's interesting.
It would be interesting. And if someone knows an example, maybe like a mutant that regulates this, you're also welcome to
Yeah.
type it here. I think that would be an interesting example to
Yeah.
hear about.
Yeah. It looks like there's a couple questions about de-differentiation. Perhaps we could touch on that a little bit. The first one is from Ramiro, who says, you said at the beginning that you don't like the word de-differentiation.
Yeah.
So
Yeah.
can you say a little bit more about that? And in particular,
Well,
this concept is quite often used in model systems of de novo organogenesis, like lateral root development.
yeah, because lateral roots develop, well, I mean, I could use that example, but they develop from particular paracyclic cells and they grow out through the tissue. Well, that's a continued differentiation. That cell it differentiated what if it's a pericycle cell where it is in position. And then it continues to go down the road and become a lateral root. Not every one of them does that. It doesn't mean that it recalled all the steps to be meristematic. It actually was a meristematic cell upon one time. So what I'm saying is it implies that you went backwards
Mm-hmm.
at your original state. I'm saying they don't go backwards. They just keep going forward forever.
They go in a new direction.
Yeah, because it's totipotency.
Mm.
It's expressing your ability to become anything you want, in a sense.
Dennis, there are actually many, many questions that we'll probably not be able to answer all of them. We still have time and we'll continue talking, but maybe just for the people that were asking and are curious about your answer for those, maybe we could collect them and if you want, send them to you. And if you
Yeah,
want, you can answer some of them and we
that's
can
okay. Yeah.
with everybody. Another question from an anonymous attendee. Does a cell need to have just one type definition? Could there be a functional structural definition and an evolutionary phylogenetic definition?
Wow, I really like that question.
Yeah, that's a good
I've
one.
never thought about that in a sense. Yes, probably so. I mean, I can understand that, or maybe it's all part of the definition. But if you're a plant anatomist, you're going to stick over into your typological side because that's what you do. If you're a plant physiologist, you're going to stick over into your functional side. If you're a developmental person, you're going to go over there. So I have to think about that. I mean, you're going to send me that one, right?
Yeah, we can send you all of them, and we can also note
Yeah, I need
what
to
we've
really
already
think about that
answered.
because of because I guess what I'm saying is anatomy has multiple purposes or serves multiple functions, right?
Mhm.
Foresters have a particular function that they care about.
I was trying to think, come up with an example for where that would have contradicting definitions. That if,
Yeah, that's what I'm
yeah, yeah. We'll
trying
think
to say.
about
I
it.
have conflicting terminology here. The hydroids versus trachary elements is one. We have two terms for things that do the same thing.
Yeah, so like same function,
Yep.
different
The leptoids are the
cell
best.
type.
Same,
Yeah,
Mhm.
same development, same function, same place in the plant. It's interesting.
it is. Yeah, it is.
I want to bring up this question from Luigi, who says, "Nice talk. So we need descriptors, and for something like a plant cell atlas, what is like? How can what are your opinions, Dennis, on how we can standardize these descriptors? What are the best ways that we can represent cell types from these new things like genomic studies?"
I suspect that, the ontology here or the atlas thing will have the what do I call it? The typology type description that we need, but within that you can include the function or the other things involved in it. So you get more information about it.
Mm.
In other words, if I was an anatomist, I might get curious about something. So I go over and I look at the physical description of this or the typology, and then I can go in there and say, oh, and look, this is what the function of these cells is. Well, that's been demonstrated. And here's the genes that led to the differentiation of that.
Hmm.
So
So
I
this is
think,
what it is, what it does, and also how it got there. Like, what
yeah.
are the genetic
So I'd like to
programs?
be eclectic about things
Hmm.
in that sense. Yeah. I think it's a good point.
Based off of this answer, I'm thinking, how do you see the future? Like, yes, we try to go as wide as we can in terms of definitions and how we look at things, but like you mentioned before, if I'm a plant physiologist, I would have my own perspective, and if I'm a geneticist, I would have a somewhat different perspective. And if I do evolutionary biologist and biology and so on, how do you see the future? How can we improve our ability to look at cells, tissues, organisms, better understand them, better define them? Although we have this certain expertise that we work on, like how is it communicating between us with the different expertise? Is it creating large resources? What do you think about that, about the future?
Well, one of the things, in my lifetime career, we got rid of botany departments.
Yeah.
They became biology departments. And then the zoologist would say, well, we don't need another botanist when someone retired or whatever. But what I'm going to, because I was in botany departments. And the one thing about if you have a plant science departments, then you can have people from these different disciplines in the same department. And if they're rational human beings, they will communicate with each other. And in
Yeah.
fact, at Ohio State, where I did my undergraduate and master's, those guys actually coordinated their courses so they could
Mhm.
build on each other. It was a very collegial atmosphere, and it worked very, very well.
So I think we a little bit maybe went backwards in that sense.
That's what I'm saying, because
Something.
Davis was the same way when I did my PhD at Davis. But Davis had so many plant scientists,
But still, I think
was-
Davis specifically still does.
yeah, the American Journal of Botany used to put a membership list in one of the issues, and Davis had 2 pages.
Hmm.
But these people all work together. So on my PhD committee, I had someone from range science and someone from horticulture
Hmm.
doing developmental plant anatomy. So yeah, I think that's what we need. We need to get back that we're working on plants and we're all got the same goal. It's to understand life on Earth and how it got to be here.
And
Yeah, I think maybe that's, one of the most exciting aspects of initiatives like this Plants All Atlas is really,
Yep.
Even if we're not all at the same place, to really rally around these ideas.
Yeah, I think that's great.
Trevor, maybe we have time for one more question.
So let's take one from the Padlet for our last question. So there's a question about cell types in 2 different species. So do the cell types in 2 different species have the same identity, assuming the same morphology and same evolutionary origin? For example, comparing root cells. So how would you think about and compare, and what can we learn from this kind of analysis?
I think I would say yes to that if they have, we know enough about their development and we know where they are in the evolutionary tree of life and they're in the same, so they have common ancestry, then probably things are going to be common and it's testable, right? Because it becomes a testable hypothesis. If you say 2 things are not the same, that's a testable hypothesis. If you say they're the same, that's also a testable hypothesis. So I would say, your assumption on the front end probably would be, I'm going to accept that as true because I don't have enough time in my life and I want to go look at one of these disparities and work that one out. I don't know if that was a good answer, but that's the way I would approach it.
That's certainly a logical one. Well, thank you so much, Dennis, and thanks everybody
Oh.
for coming. I want to mention that our next seminar will be Rachel Shahan from UCLA and Jeff Scheibinger from UBC, who are going to talk about acquisition of cell identities, and that's going to be on December 10th at the same time, 9 a.m.
Great.
Pacific. So thank you, everybody, and we look forward to further discussions.
Okay, thanks.
Thank you so much.
Yep. Bye.
Bye-bye.